Thank you, Mr. Daes, for that comprehensive discussion. Again, I am Walamay Katarina and I am here to discuss the remaining subtopics, which are the advantages and disadvantages of factorial treatment structure, as well as the interaction. To begin with, let me first present to all of you the advantages. First, the most efficient use of resources. In factorial treatment structure, all experimental units are utilized in evaluating effects, resulting in the most efficient use of resources. Second, increased range of condition tested. So, in factorial treatment structure, the range of conditions tested can be increased with a minimum outlay of resources. Third, the interaction between factors can be estimated. And fourth, a factorial structure of treatments is optimal for estimating main effects and interactions. So, this means that even if the data do not come from an experiment, but the grouping factor is arranged into all combination of some other factors, then we can still say that the groups have factorial treatment structure. As an example, suppose that we measure blood pressure in 6 groups of people, which is from male children, female children, male young adults, female young adults, male middle-aged adults, and female middle-aged adults. The factors of gender and age are not randomly applied, but we can talk about the 6 groups having factorial treatment structure in these 2 factors. Initially, we will consider models for data where only 2 factors are used. And also, we will at first only consider balanced data, in which all treatment combinations have the same number of n of replicates. Moving on, we have here the disadvantages. First, large numbers of combinations are required. So, in factorial treatment structure, very large numbers of combinations are required or needed to study several factors at several levels. Second, the large size of replicates decreases the efficiency of the experiment. And third, reduction of the size of the replicates by fractional replication or other means makes statistical analysis more complex and can create confounding. Along with testing the effects of 2 treatments in a factorial treatment structure, it is possible to test for the interaction effects of the 2 treatments whenever multiple measures are taken in each cell of the design. So, interaction occurs when the effects of one treatment vary according to the levels of treatment of other effect. Interaction is defined as the failure of one factor to give the same response at all levels of the second factor. Factorial treatment arrangements allow assessment of the interaction between 2 or more factors. Such an assessment can be very informative. When significant interactions are present, the interpretation of the main effects must be modified in the light of the interaction. It is not logical to make general statements about the main effects when significant interactions are present because it is apparent that one factor does not give the same response at all levels of the other factor. Therefore, the experimental results must be interpreted in terms of the interaction between the factors. For example, in a study examining the impact of temperature and humidity on a manufacturing process, it is possible that temperature and humidity will interact in such a way that the effect of temperature on the process varies with the humidity. Low temperatures might not be a significant manufacturing factor when humidity is low, but might be a factor when humidity is high. Similarly, high temperatures might not be a factor with low humidity, but not with high humidity. As another example, suppose a business researcher is studying the amount of red meat consumed by families per month and is examining economic class and religion as 2 independent variables. Class and religion might interact in such a way that with certain religions, economic class does not matter with the consumption of bread meat, but with other religions, class does make difference. In terms of the factorial treatment structure, interactions occurs when the pattern of cell means in one row going across columns varies from the pattern of the cell means in other rows. This variation indicates that the differences in column effects depend on which row is being examined. Hence, an interaction of the rows and columns occurs. The same thing can happen when the pattern of cell means within a column is different from the pattern of cell means in other columns. Interaction can be depicted graphically by plotting the cell means within each row and can also be done by plotting the cell means within each column. The means within each row or column are then connected by a line. If the broken lines for the rows or columns are parallel, no interaction is indicated. As you can see on figure 11.10, it is a graph of the means for each cell in each row in a 2x3 or 2 rows and 3 columns factorial design with interaction. Note that the lines connecting the means in each row cross each other. In figure 11.11, the lines converge, indicating the likely presence of some interaction. Figure 11.12 depicts a 2x3 factorial design with no interaction. That is our discussion all about. Since this topic focuses on the concept of factorial treatment structure, make sure to stay tuned for the next reporter as they are about to discuss on how to compute two-way ANOVA using factorial design. We hope that you learned something from your study buddies. Thank you for watching!